A new study finds that nearly 40% of the human genome remains largely unexplored, which could dramatically impede the search for new drug therapies. The dearth of research tends to be self-perpetuating, because scientists know they are more likely to be successful and funded by querying those areas of the genome that are already relatively well described. As a first step toward solving the problem, the team of U.S. and European scientists has proposed an elegant method for categorizing the genome according to how extensively genes, and the proteins they code for, have been studied. The new classification scheme also suggests what steps would be needed to establish whether these proteins are likely targets for an array of novel drug therapies. The work was carried out as part of the Illuminating the Druggable Genome (IDG) project, the National Institutes of Health Common Fund initiative. The findings are highlighted in Unexplored Therapeutic Opportunities in the Human Genome, published in the February 23, 2018, issue of Nature Reviews Drug Discovery. Almost 9,000 proteins are not currently being subjected to NIH-sponsored research, said lead author Tudor Apria, MD, PhD, professor and chief of the Translational Informatics Division in the University of New Mexico's Department of Internal Medicine. This study attracts attention to that situation. We hope to encourage scientists and funders to explore the unknown. Apria led a team of researchers from the IDG Knowledge Management Center in an exhaustive review of a wide array of genomic, proteomic, chemical and disease-related data in order to track the target development level (TDL) of human proteins. The researchers proposed grouping human proteins into one of four TDL categories. TClin for clinical represents the most studied proteins, ones that have known interactions with approved drugs and for which there is an identified mechanism of action. These represent just 3% of the human proteome. CHEM for chemical includes proteins that are known to bind to small molecules with relatively high potency. This group accounts for 6% of the proteome. T-biobiology refers to proteins for which there is experimental evidence of disease relevance, and some understanding concerning their structure and function, but which have not been fully developed as drug targets. About 53% of the proteome belongs to this category. T-dark referring to the dark genome includes all proteins that do not meet the criteria for inclusion in any of the other categories. T-has are proteins for which there is the least current knowledge and a low number of specific molecular probes available, the authors said. Some represent unexplored opportunities within the druggable human genome. These proteins account for 38% of the proteome. The authors note the funding to study the dark genome has been scarce, in part because research is often based on artificial distinctions, in which problems are segregated by organ toward disease. This distinction breaks down in nature, as we are likely to observe the interplay between the same genes and pathways regardless of organ, albeit in the context-specific manner. Their proposed classification scheme provides a convenient way to partition human targets that highlights the focus or lack thereof of science and drug discovery efforts on different targets. Apria said the fact that drug targets for which small molecules are currently known represent only around 10% of the human proteome underlines the extent to which we don't fully understand human biology. There's a lot more work to be done. Background: the Knowledge Management Center includes researchers from the NIH National Center for Advancing Translational Sciences, the University of New Mexico, the University of Miami, the University of Copenhagen, the European Bioinformatics Institute and the Icahn School of Medicine at Mount Sinai. Other collaborators on this paper are from Yale University, the University of North Carolina, the University of California, San Francisco, Baylor College of Medicine, the University of Zurich, as well as IQVIA and AstraZeneca.